Good afternoon, everybody. My name is Juan Bueren. I am the current Vice President of the European Society for Gene and Cell Therapy. And together with our President, Dr. Hildegard Büning, we would like to open this important series of e-schools that we have launched as an alternative of the spring school, which unfortunately could not take place due to the COVID pandemic. Well, to start with, I am of course very pleased to introduce Professor Büning to you. She serves as current president of the European Society for Gene and Cell Therapy, and also as scientific secretary of the German Society for Gene Therapy. She is also professor of infection biology and gene transfer, and is a deputy director of the Institute of Experimental Hematology at Hannover Medical School in Germany. Hildegard studied biology in Münster and in Munich, and she obtained her PhD at the Ludwig Maximilian University in Munich in 1997. Since then, she has been very active in the field of adeno-associated virus vectors, with a very particular focus on AAV host interactions and AAV vector development. And she is nowadays developing strategies to modify vector tropism and immunogenicity of these vectors, using both rational design and library-based approaches. So we are lucky that today Hildegard will introduce what ESGCT is, and also she will open this e-school series with an overview of the current status of gene therapy. To conclude, after her presentation, you will be able to ask your questions, write them, and I will raise the questions with Hildegard. So thank you very much. Enjoy the lecture, and it's your time, Hildegard. Thank you.
So thank you very much, Juan. I'm just trying to start my presentation. Yes, wonderful. So also from my side, welcome, and also of the whole European Society of Gene and Cell Therapy. And before I let you know a little bit more about from concept to proof with regard to gene therapy, I would like to invite you to follow me a little bit on what European Society of Gene and Cell Therapy is. So we are a non-profit organization, and we aim to promote basic and clinical research in gene therapy, in cell therapy, and genetic vaccines. And as you all know, in the time of COVID, in particular, the latter is very, very important. And how do we do this? We try to facilitate education, and this e-school is one kind of how we do it. We exchange information technology, and we would like, and we do, serve as professional advisor for stakeholders and regulatory bodies. And so what we do is let me see. I have a problem with my screen here. So, okay. So it's always a technique, you know. So the ESGCT was the idea of having the society launched was starting only 7 months after the first human gene therapy trial. And I will tell you a little bit more about that. So back in 1990, this trial was started, and these names listed here were the brave person saying, we have to do really something because of these fascinating ideas and which are lying in front of us. And so they were initiating the international workshop. And honestly, this is a time when I was still a student, so long time ago. And only 16 months after the first gene therapy, so really, really short thereafter, the society was founded as a working group on human gene transfer and therapy. And here I would like to point again out to the persons who were the first doing this and really initiating our society. Then in 1998, we changed the name into European Society of Gene Therapy, and then due to the advancement which everybody sees if you use gene transfer to put new features into cell, we changed the name again into European Society of Gene and Cell Therapy. We have our annual congresses once per year. And as you see from these nice pictures, we are traveling through Europe. And this is something which is very important for the society, because we really would like, and we do, closely connect with the different national societies in Europe working on gene therapy, because we think when we work together, and this is really what is gene and cell therapy is about, this is how we get this fascinating and very powerful technology and treatment strategy to the patients in need. It's really an endeavor which we have to do together. We do have our own society journal, Human Gene Therapy, in which our abstracts are published, and also we always have a special edition around the national congresses and the annual congresses, so look into that. And then, as Juan already told you, we have a special lecture course which started in 2016, which is absolutely dedicated to PhD students, MD students, and MD PhD students. And it's in a congress-like format where we are together with 150 students 3 days and talk about science, have a lot of fun, and it's all about networking and education. And as I told you, we do have once per year an annual congress together organized with national societies. And these are just some pictures of our last congress in Barcelona. And just to give you a flavor how this looks like, and to let you know that with our 1,254 members, it's really that we do have members from all over Europe and beyond. So this is new now, the eSchool series, which we started due to COVID. And we thought this would be good and a nice opportunity to really do education. It's open to everybody. And these are the next sessions, which will start focusing on viral and non-viral vectors for gene therapy and gene editing. And as you see from the names, these are really experts in the field. And then we move on, we will tell you how AAV vectors and lentiviral vectors are produced. We will tell you what you should look at when it comes to the genetic payload. We will discuss aspects, safety aspects on gene therapy, but also on genome editing and gene editing. And we will have detour to different gene therapies which are ongoing and which are in preclinical development as well. So we try to cover all the different aspects in cell and gene therapy as we do on our national congresses, annual congresses, and as well as our spring school event. And then we also would like to launch very close a so-called e-seminar where we will focus on hot topics. And of course, at the moment, COVID or SARS is the one everybody is speaking about. And if you see also here the names, we are supported by very well-known people who will speak and discuss different aspects with you. And also here, as the e-schools and the e-seminars, it's really that you can also launch questions, which we try to answer then afterwards. So after this little introduction to the European Society of Gene and Cell Therapy, I really would like to kick off by letting you a little bit know on the background, so about gene therapy from the concept to the proof. And then, as I said, the next webinar will be focusing then on the delivery tools and so on. So now come with me into gene therapy from concept to proof. I really like to start with this slide, this quotation is from Luigi Naldini, who is the director of San Raffaele Telethon Institute for Gene Therapy in Italy. And by the way, he was also president of our society from 2012 to 14, because he really summarizes what fascinates clinicians and scientists and the general public when you name the word gene therapy. And this is because its potential to treat a disease at its genetic roots. This is achieved, as you know, by counteracting or replacing a malfunctioning gene within the cell that is affected. And this you can really only do by gene therapy. And when you look now into cell therapy, then by the technologies which we use in gene therapy, you can turn these cells into smart vehicles. So gene and cell therapy is tightly connected. So to very simplify the concept, is really we have a gene vector, a gene ferry, we have a transgene. We need to know, of course, the target where the ferry has to go. And then we should have a therapeutic effect. And in principle, you can use a transgene in addition to what is present in the cell, which we call gene addition therapy, or you go and replace the malfunctional gene by the new one. As you can imagine, and as you will see when we go back in time, gene addition was a way to go so far because the tools were not available to precisely replace a malfunctional gene. This, of course, comes to the cost that we face a problem that the malfunctional gene is still there. And also, if we cannot replace but add a gene, that we cannot really use the controlling function of the environment of the chromosome and the being in the right location and so on and so on. But what delivery tools do we have in hand? And you see, I start with saying non-viral vectors. So you can assume that the other one will be a viral vector. So 19% of the gene therapy application use non-viral vector, and the most easiest non-viral vector is, of course, naked DNA or plasmid DNA. And when you go back in biochemistry, then you know that your DNA molecule is negatively charged. And when you think about how your cell membrane is charged, it's clear that you need to do some sophisticated tricks to get the DNA into the cell. And so to help this a little bit, scientists came up with the idea to cover the negatively charged DNA by, for example, cationic lipids or cationic polymers, so that by this you can simplify the transfer into the cell. And then, of course, the nice thing which you have in hand here as synthetic vectors, you can equip them with ligands, allowing you to bind this polyplex or lipoplex to the cell, and also with functions allowing them to be released from the endosomal compartment and go straight to the nucleus. So as already discussed, besides non-viral vectors, we have, of course, also the viral vectors. And on one hand, we have those which are based on an RNA virus. And you may all know about retrovirus and lentiviral vectors or viruses. And these do have RNA, which needs to be transferred into a DNA before it's integrated into the host genome. And this is quite important because this integration function is advantageous for a lot of different applications. On the other hand, we do have the DNA viruses, and I would like to name here only adenoviruses and adeno-associated viruses. And these do have a DNA, which they deliver as a cargo. And both of these viruses, at least as a vector, do not possess an integrase. So in this case, it's not kind of a life cycle as a vector to integrate. And so other application, of course, are the targets if you would like to use vectors which are based on AAV or adenovirus. If you just look at the numbers, so the percentages, these viruses are used as vectors in gene and cell therapy, you see that a lot more applications use viral vectors, which, as I said, are based on these viruses. And why is this so? So why do we like to use viruses? This is because nature invented sophisticated and very efficient strategies to deliver genetic material into the cell via viruses. So, and if you remember what I told you with the non-viral vectors, the field of non-viral vectors really covers these strategies to really get this non-viral vectors into the cell via receptor-mediated endocytosis to be released from the endosome and so on. But this nature has already equipped a virus with, because otherwise this virus cannot survive. And if we really put it down into the basics, then a virus is nothing more than something like a protective coat, protecting the nucleic acid from the environment and also from components in the blood, in the tissue or whatsoever. And of course, the genetic payload, which is delivered within the virus, within the protective coat. And there you find all the information which is specific to the virus and cannot be covered by the cell, and which allows then the cell, under the guidance of the virus, to generate new virus particles. However, we don't want to use a virus, at least in the classical sense of cell and gene therapy. What we need is just the features which nature has equipped a virus with to deliver the genetic material. And so we would like to exchange the genetic material from a virus to something which we would like to deliver, namely our transgene. And what we have to do therefore is we have to have the transgene either in an RNA or a DNA format, as depending on the vector system you are using. And we have to equip these nucleic acid with a specific sequence, ITRs, LTRs, or something like that, that it's clear that this one belongs into the respective protective code. And then the viral vectors will interact with the cell as the virus has learned through evolution. How we really generate these vectors, how you have to do the purification, which things you have to look at if you would like to design your vectors, this you will hear about in further webinars, which are in this series. Now you do have your vector, you do have your transgene, which you would like to be delivered into the patient. And now the question is how you get these genetic material into the patient. And in principle, you do have two routes of application. In one hand, you isolate from the patient's cells, such as hematopoietic stem cells. You put them in culture, you modify them with your viral vector of choice so that in at least part of these cells, you do have your transgene present. And then you give this back into the patient where these modified cells will engraft, for example, in the bone marrow and give rise to progeny and descendant cell types, which then, for example, as you will see later in my talk, for example, will rise the immune system. On the other hand, you can, of course, also apply your gene delivery tools together with the transgene, of course, directly in the patients. And here you have obviously two different choices. You either put them locally, so for example, into the muscle by a syringe, or for example, you apply them through a vena, so intravenous administration. Now let us go back in time. And I would like to start, as I was promising you already, in 1990. And what was going on there? So if we go to UK, Margaret Thatcher was still Prime Minister. George Bush was President of the US. And in Germany, Helmut Kohl was there. We just experienced the fall of the Berlin Wall. And if you look at the Nobel Prize in medicine, this was given to a team who worked on transplantation. We have the Hubble Space Telescope, which was transported by the space shuttle at that time into the orbit. The Human Genome Project was launched. So this is the time where we now have to stop to start with our view on gene therapy. And the disease which the first gene therapy trial in humans was focusing on was adenosine deaminase deficiency. And of course, although I said we start in 1990, a lot of work had to be done before. One had to generate and to learn how from a virus you can do a vector. You have to learn how to cultivate cells. You have to know how to do a transplantation and all this, a lot of things. So to learn to clone and so on and so on. All this already helped on the route to gene and cell therapy. But now back to adenosine deaminase deficiency, which was the disease which the first human gene therapy trial was on. So this disease, if you look in the internet, you find that it's a 1 to 200,000, an incidence of 1 to 200,000. It's autosomal recessive. And you see that it is an enzyme, and this enzyme is required to distract ATP and DATP. And so you can imagine if this enzyme is not functioning, cannot do the deamination of adenosine and deoxyadenosine, then a metabolite will accumulate. And basically this metabolite is quite toxic, and toxic to different organs, but in particular sensitive are the lymphocytes. And therefore, if you look into patients suffering from ADA deficiency, you see that they show up with a severe combined immunodeficiency. That means they don't have T, B, and NK cells. And so as a clinician, you see that these little children, these babies, come up with infections. Knowing that you do have a stem cell and that from stem cells, you can build up the whole immune system, one option to treat these patients is, of course, bone marrow transplantation. And this at that time was also not that easy to do. And of course, if it is an allogenic one, you face a problem of that maybe the matching is not 100%, you develop a graft versus host disease. So the team of Michael Blaise and French Anderson and a lot of members which are put here on that paper, which you see, were brave enough to say that why, if we don't have, if we have the tools, if in principle we know that this is safe, why shouldn't we help these patients by modifying their own cells? So gene therapy was launched. The first patient was 4 years old in September 1990. And the second patient, 9 years of age, was treated 1991, so in January, so quite close. And what is important is that these patients, so of course they had ADA-SCID, but they had no donor available. and they were under replacement therapy, which allowed the team to isolate T cells. And they decided to go for T cells because in preclinical testings, they saw that if they modified the bone marrow with the tools they have at the moment, that the efficiency of stem cell engraftment was low and maybe not sufficient, while T cells maybe looked like an easier task. And what was also there already, and that is what I said, that a lot of work prior to this gene therapy was required. It was already established that you can use a retroviral vector for doing a gene transfer into cells, that it mediates a stable integration of the genes which you have transferred, and that you can use this in human cells and in humans, at least when you give back cells. And please have in mind, we are speaking now of a retroviral vector, so a vector which delivers an RNA, which is then transferred into DNA and can integrate into the host cell. And the therapy, the clinical trial could show that it was safe to do it like they wanted to do it. They saw that the ADA levels increased and that the patients got less infected by viruses and bacteria, that they reacted and had T cells. And I found this picture. So you see the two girls which were treated back in 1993. And here's another picture of the two in 2013. With the knowledge we have now, and all of you, I assume, and also with the knowledge that people had at that time, it's clear that if you would like to really get the whole immune system back, and if you really would like to have something which you can use lifelong, so a one-time treatment, then you should go to the hematopoietic stem cells to modify, because these stem cells will then engraft into your bone marrow and give rise to all the different cells. And if you did here the repair, then these cells will be fine. And so also besides ADA-SCID, also other SCID diseases came into focus, but not only for those, but also for diseases which target other cells of the hematopoietic system, or even cells which are later on involved in metabolic diseases, can be really treated by modifying the hematopoietic stem cells. So for the next stop with our time machine, I would like to stop in 1992 and a bit later, because this was then when we looked into the gene modified hematopoietic stem cells, so leaving the T cells behind and move to the stem cells. And here I would like to go to another SCID, severe combined immunodeficiency, and this is SCID X1. And in contrast to the ADA-SCID, you see that we are now coming up with a disease which is located where the gene is located at the X chromosome. And what you also see is that we don't have an enzyme, but in this case, it's a subunit of an receptor, an interleukin receptor. And so you can imagine if your immune system is going to start and to build all the different parts, how important it is that the cells can communicate, and communication is really through the receptors. And if you don't have receptors, it's clear that that you run into a severe combined immunodeficiency, because you don't have T and NK cells, and because you don't have T cells, also the B cells are not working. Also in this case, it's allogenic bone marrow transplantation, what is thought as a therapy. And so, like in what we discussed before, for these patients, it would be also good if your own cells can be modified by the gene therapy. But as I said, in this case, we are changing to the hematopoietic stem cells, CD34, not to the T cells. And indeed, this approach was done by people in Paris, so the group of Alan Fisher and Marina Cavazzano-Calvo, but also in parallel in London with Adrian Trescher and his colleagues. And for gene therapy, this was the first real report on a cure, because it really could be shown here that due to the therapy, the whole immune system went back. With the ADA-SCID, it could be shown that the therapy, the strategy was safe, but the two girls were remaining on replacement therapy. So you could not 100% claim that everything was due to the therapy. The effect was 100% due to the therapy. Here with SCID-X1, it was the real first proof that gene therapy works. And this result was quite important for us. Here is a report from the Paris trial, and here is just a picture showing you a patient which was treated in London, just to show you that these patients are really, after the therapy, were able to go out. Before that, every breath which they would have taken without sterile air would have caused infections. And these first cure and this really proof that the concept holds true was very important because of something which happened here in the gene therapy trial on ornithine transcarbamylase deficiency. In this case here, it's a disease which is not in the hematopoietic system, but it's a disease which affects the urea cycle, because you cannot change this molecule into citrulline. And therefore, you accumulate ammonia, and this is, of course, not healthy, resulting in coma, brain damage, and death. So the trial which was designed here was using an adenovirus as a vector, delivering the defective gene in a correct version, and it was directly applied into the hepatic atria of the patient. And why I'm mentioning this and why I'm saying that it's so important that we had in 2000 the report on the real first cure is because in this clinical trial, the first death due to gene therapy happened. So this boy was 18 years of age. He was in the high dose group, so he received 6 × 10⁻11 particles per kilogram body weight. And what happened was that he reacted with systemic inflammatory response, and this could not be stopped anymore, so he died. And in this paper, Raper and colleagues went back to the samples and tried to understand more what happened. And I copied in here from the discussion 2 things, which I think is quite important for us also if we now design gene therapies. So what they say is that the toxicity which was observed was not the toxicity which was due to the adaptive immune system. You know, we have the innate and we have the adaptive immune system. But what their results show is that it was due that the vector was selectively targeted to antigen-presenting cells in the liver and spleen, and therefore activating the production of cytokines, and really then this triggered the massive immune response. So based on these results, if you would like to really change this, you have to change the capsid, because this is, so the outer shell, this is the thing which was recognized by the immune system. And the second part, which I here also highlight, is that they typically say that you really have to look at the model system you are looking at, because what you see here is that the efficacy with which the vectors delivered their transgene to the hepatocyte differed not between human and the preclinical model, but the severity of toxicity, so how the host reacted, is totally different. So that we see that the mouse was, and also the non-human primate, were much more resistant to the activation of the innate immune system than the human. This already shows you how important it is to have the right model, and also how difficult it is really to start new therapies. So when we now go further, then also with the primary immunodeficiency, although it is clear and showed by the SCITX1 trial that the underlying genetic defect could be treated, in 2003, this news came up, which shocked also the whole community, because leukemia was developed in a patient. And also this was the Paris trial. Also in the London trial, one of the 10 children which was treated developed a leukemia. So if we have a look at the time until now, and in particular also around 2002, 2003, then we see that ex vivo gene transfer with using a gamma retroviral vector, which integrates the therapeutic gene into the host genome, resulted in a clinical benefit for the patients. So you can overcome the primary immunodeficiency and regenerate the immune system. As I showed you in this example of ADA-SCID, SCITX1, also ADA-SCID was further developed, and it could be shown that indeed the therapy is curing the disease. Also for other diseases like CGD or WAS, it was shown that the therapy works. However, what was also observed then in 2002 and the following, that in some of these diseases, the leukemia is induced. And it was seen in SCITX1, where 6 of the 20 patients developed a leukemia, in CGD and in WAS. But it has never been observed when treating ADA-SCID patients with a gamma retroviral vector. So what I told you here is bringing us to a challenge if you would like to modify hematopoietic stem cells, because on one hand, you need to have a stable modification. You have to integrate your transgene into the genome of the host cell, into your stem cell, to really cure the disease, because from the stem cell, the T cells and the B cells and the NK cells are coming from, and you really need to have the cure done there and to have it stable. But on the other hand, at that time point, you are lacking precision tools. And also at that time point, lack of knowledge of certain features of the gamma retroviral vector was there. So the gamma retroviral vector, yes, everybody knew that it integrates, because it's a kind of the normal life cycle of the retrovirus to integrate. But what came to light is that it is not a total random integration, which would be less difficult to handle, but it's a semi-random one. So gamma retroviral vectors like to go into control elements, which is might be of danger if you integrate your transgene there, and thereby may disturb neighboring region. And also the copy number that was used, so the amount of vectors were quite high. The regulatory elements, so driving the transgene, was quite high, and the disease context was important. Because what induced the leukemia was that the gamma retroviral vectors landed nearby a proto-oncogene and turned it on. And I named here the disease context because, as I said, with ADA-SCID, although the same vectors are used and the same integration sites are used, it seems to be safe. due to what we learned and what gene therapy learned with these cases, the vectors were modified, the integrating systems were optimized, so the gamma retroviral vectors were changed so that the LTRs are not anymore the promoters. We have an internal promoter and so on and so on. And we changed also from the retroviral vectors to the lentiviral vectors. This is all I would like to tell you about these. Of course, you will get a special lectures about the retroviral vectors, insertional mutagenesis, what was learned about these early trials, and now how the new vectors are designed and why they are safer by Axel Schambach in the next seminar. However, what I would like to tell you is that in 2009, these new vectors, so those based on the lentiviral vectors, were first applied in a human clinical trial for a disease which is called adrenoleukodystrophy. And in this case, it's not a primary immunodeficiency, but a metabolic disease. And it's important that the modified cells will also move then after being reinfused to the brain. So again, CD34 cells were the cells which were ex vivo modified, this time with the next generation vectors, so lentiviral vector, and then reinfused to the patient carrying the respective transporter. And this is again highlights the report, and it was done by Nathalie Cartier and Patrick Aubuch. And Nathalie was also president of our society not long ago. It was in 2014 to 16, so quite close in time. And the nice thing is that these therapies, so you have to imagine, before that we knew that gene therapy, ex vivo gene therapy, reinfusion of the modified cells would help the patient, but there is a risk of gene therapy, the risk of leukemia. And now is in the test of the new vectors, of the lentivectors. And it's a real milestone to see that these vectors really helped to stop this very severe disease where you have a demyelination. This is the brain of the patients where you have a demyelination disease. And the patients which received the ex vivo modified cells do much better. You have a stop in compared to patients without gene therapy treatment. The next stop I would like to do is in 2008 for congenital blindness disease, where patients suffering from this disease know that before they will come to the age of 30, will lose their vision. And here we report about a vector, the adeno-associated viral vector, which is applied in vivo. So we leave ex vivo and go in vivo. They are applied in vivo locally, subretinally. Really, it's a needle which puts the virus to the right place in the eye. And it was done in 3 clinical trials in parallel. And it was not only shown that we had safety in these clinical trials, but also efficiency of these clinical trials were shown. Also, acquired inherited diseases can be treated with gene therapy, of course, and one of a very what is not rare, of course, are cancers. And in 2011. Carl June was first reporting on the efficacy of modifying T cells by chimeric antigen receptors. And these molecules, which are synthetic, artificial molecules, these molecules now allow the T cells to find specific molecules on the tumor cells, which I otherwise would not find, and then result in the killing of the tumor cells. And you know that in particular, the CAR T cells and CAR NK cell field is exploding nowadays. Also in 2011, another milestone was reached, namely not a local injection of an AAV vector, but an intravenous administration of a vector of AAV, which then accumulated in the liver as it should be, and there was producing a factor allowing to treat patients with hemophilia B, so factor 9. And the nice thing here is really that it was possible to really use the natural tropism of the vector to just use an IV infusion to get the vectors to the right cells, the hepatocytes, which should express this factor. And still these patients show that a benefit from this therapy. So they are off replacement. The next stop of our time machine is 2012. And why is this important? This year, as for gene and cell therapy, quite important because it's the first time that a gene therapy has received marketing authorization in the Western world. And again, it was an AAV vector, which was used for an ultra rare disease. So you have seen a rare disease, you have more common disease, and now the first approval was for an ultra rare disease called lipoprotein lipase deficiency, and where the vector was delivering the transgene in a gain of function version, and it was put really into the muscle. And the difficulties here was really to have enough patients to really show that this therapy works. However, it was approved and it was a breakthrough. And I just put here some of the newspapers, and it was really like that the first time also the regulatory agencies had to make the step into, or decided that we were at a stage that gene therapy can go to the next stage, leaving the clinical trials and become a drug. Glibera, this is the official name, however, is off the market already. And after the approval, only one patient actually in Berlin was treated, because on one hand, it's an ultra rare disease, and on the other hand, because of the cost of this therapy. However, because it all started, the next approvals were following. So in 2015, there was HSV-based oncolytic viruses, so herpes-simplex virus-based oncolytic viruses in the field of cancer. You see 2016, I listed ADA-SCIT, and you see in 1919, the first human clinical trial on ADA-SCIT was performed. First, these patients were still left on enzyme replacement therapy, just to be sure, but in the later days, this was removed, so really that the therapy given by the modified stem cells were the one resulting in the cure. And these results then resulted in the marketing approval for Strimvelis, so gamma retroviral vector ex vivo modification of hematopoietic stem cells. And then a year later, approval came for the CAR T cell world, so Kymriah and also others. And then we have in 2017 also the therapy Luxturna, which is an AAV vector, and you saw me mentioning the AAV2 subretinal injection for the treatment of inherited eye disease, which was liver congenital amaurosis. And this therapy is indeed for those patients. So if we now just have a look. So we started off with the first human clinical trial in 1919. And when we stop in 1919, we see that we have 9 marketing authorization of gene therapies. And if you look into press, you see that, for example, the Food and Drug Administration in US expect really from 2025 onwards that up to 20 new marketing approval per year will come up. This already shows you where we are at the moment, showing that gene therapy is now really showing that the concept works, that patients benefit from the treatment, and that we really have drugs approved which can now go into the patients. However, there are barriers towards making gene therapy a clinical reality, or really for bringing gene therapy to the patients in need. Before, I would say 2012, the first marketing authorization, we had the challenge that gene therapy vectors the efficacy had to be increased, and so on and so on. We really had to prove and to show that the strategies are working. And now we are tackling other barriers, and it's still an individualized therapy. The costs are extremely high of the therapy because at the moment it's rare diseases, highly sophisticated, and so on. We have still the question if the safety data are sufficiently solid to expand beyond rare diseases and cancer, so to go to more common diseases. How should we control the misconduct? When do we start treatment? So childhood, earlier. How does gene therapy impact on the patient's life? How do we monitor or do the following up even when we give it as a drug? So how do the clinicians do this? We face maybe the bottleneck of vector production. And at the moment, for most of the therapies, it's really that we have to do them in specialized centers. And by this, I would like to stop, and I'm happy for your questions. And just I would like also to announce and remind you that the first scientific contribution in our e-school will be from Axel Schambach on gamma retroviral vectors. Thank you.
Thank you very much, Hildegard. Thank you both because of the very nice introduction of the ESGCT. And by the way, I would like to take advantage of your presentation to invite to all this audience to enter into our society and to be members, active members of it. And also thanks very much for this very wide and deep overview of gene therapy in the past and in the present. And it was very clear the main problems that gene therapy had in the early times. And it was extremely interesting to be part of those times in which the severe side effects generated as a consequence of the first gene therapy vectors have been corrected very significantly with the development of the new vectors that you have mentioned. And that in fact nowadays, none of the more than 200 patients that have been treated with new vectors with inactivated enhancers have developed any significant side effect. So thanks very much for all of this. Now, while I receive some questions from the audience, I would like to ask you for a general comment, mainly focused for the young population of students that are attending our e-school. How do you think that gene therapy will evolve in the future, and which of the different aspects of gene therapy you think will need for more specific and deeper basic research, that should be conducted by new generations of investigators?
So I think we have so far proof that the vectors which we have in hand are capable of transferring the transgene into the cell, that we get a clinical benefit with the system we have. But still, we have to apply a lot of vectors, be it ex vivo to modify the cell there, or being in vivo. And so what would be very important is that we enhance the efficacy of the system so that the vectors are becoming more efficient, more specific, so that on one hand, the production costs go down because we have less vectors which we need. And on the other hand, that we have less antigenic load for the patients. And also you have to imagine when we put the vectors into the patients that we need to have, that the immune system will react, and that it would be very good to have them really only when you do it in vivo, specific for your target cell, to really minimize also the reaction of the immune system. So this is one hand. But on the other hand, we can do a lot by really understanding the disease and see the and develop the vectors in particular for the diseases, that it's really adapted, that we, for example, understand the differences between ADS-KIT and X-KIT. These are all things which are very important. And of course, we need to understand what we do to the cells if we do the modifications. And this is just for the gene addition. But if we now go to the gene replacement, what we have now, due to the technological developments, the option to really correct the defect gene by replacing it. So we have to further develop the specificity of these tools, the efficacy of these tools, and to understand how the natural regulation at the site where we integrate these transgenes occur, just to make this also quite effective and safe for the patients.
Thank you, Hildegard. Now we have 2 minutes and 2 questions, right? So the first one comes from Antonio Liras, who asks you, almost all current studies are based on gene therapy. Are most of them combined with cell therapy?
So if you go for ex vivo modification, then it's a combination between gene and cell therapy, because the gene transfer into the cell, there you use the same methodology you would do for gene therapy. So if you do ex vivo modification and back this as cell therapy with modified cells.
The next question for you, although this could be also part of Axel Schambach seminar, comes from Kali El Badri, and she asks, I am interested to conduct PhD in gene therapy research. Any tips which I should consider when looking for a position, and how to find the main corresponding research organizations? And okay, there is a third one. So you have 30 seconds for this.
So how to connect to find a research organization where you apply to?
Yeah.
Just send us an email. We will help.
And be part of the society.
Yes.
And from Tanja Hirsch, this is the one that will be related with insertional oncogenesis. Is it clear nowadays what exactly is the reason that there are no leukemias observed in ADA-SCID trials compared to WAS or X1-SCID?
So this is a question Axel will discuss, but it's the disease relation. So it's the condition of the immune system is different and the thymus and so on. So it's disease related. This is what at the moment is discussed.
The very last question. Now questions are coming. I will try to be very brief. What is the exact role of ITR?
ITRs.
from ITRs, from MUSAC MK.
So the ITRs are the packaging signal. So the signal saying, okay, this DNA has to go into the AAV capsid, and it's also serving as origin of replication.
Okay, once more from Arjun Padman Blan. When will the prohibit cost of the products for patients be overcome? It's a good question indeed.
When with regard to the costs, yeah, I mean, the problem is indeed that this has to be discussed with the agencies who are, so the insurance who are paying, at least in Europe, for the costs. So a lot of people are working on how to discuss the cost and make it better available for the patients.
Okay, so for sure that many others will come, but it's time to close. We have to be punctual. And once again, thank you very much, Hildegard. And then I would like to remind to all of you that next Wednesday, same time, Axel Schambach will give a fantastic lecture on retroviral vectors. So thank you very much to everybody, and see you next week. Bye.
Bye-bye.